Tēnā koutou katoa, nau mai here mai, ki te tēnei wānanga I Purangi. It is my pleasure to warmly welcome you all to our Taniwha webinar on avian influenza. My name is Lucia Schweitzer. I am the Porangahau, the Research Engagement Manager for Taniwha. Many of you will already be familiar with Taniwha, the infectious diseases research platform, co-hosted by ESR and the University of Otago. To those who have not had much interaction with Taniwha before, please let me briefly introduce us. Funded through the Strategic Science Investment Fund out of MB as the infectious diseases research platform, Taniwha was gifted its name based on this tongikura. Kia niwha te nākau, ki te whakau, I ngā mahi atawhai mo te iwi. This tongikura reminds us to be brave and steadfast and do what is best for the people. It is a nod that infectious diseases may present difficult challenges for many of us, but to stay steadfast in our work and always keep the best for the people in mind. The vision for Taniwha is to form a strong, prepared and unified network to address any current, ongoing and emerging disease threats together. For Taniwha to fulfill this vision, we need to achieve our mission, ensuring world-class research capability to support our preparedness for current and future infectious disease challenges. I expect that you will see prime examples for how we work towards our vision and mission in today's webinar. And for more information, please visit our website, taniwha.com. Without further ado, I will now hand over to distinguished Professor Nigel French, our esteemed Putayau, Chief Science Advisor for Taniwha.
Ngā mihi Lucy, tēnā koutou katoa. It's a real pleasure to be able to help and present this webinar today. So thank you very much for attending. Just by way of introduction, since 2020, a new strain of highly pathogenic avian influenza or bird flu has been sweeping the world, resulting in large numbers of deaths of wild birds and also affecting domestic poultry as well. Multiple spillover events into mammalian species have also occurred, including marine mammals, farmed mink, cats, dogs, and more recently cattle in the United States. A small number of human cases have occurred, raising concern about its potential to evolve into a strain that could be transmitted between people, that would then cause more widespread infections. New Zealand has never had a case of highly pathogenic avian influenza, and Oceania is the only continent to remain free of this new strain. Surveillance remains key to early detection and a rapid response should it arrive on our shores. And Taniwha has recently funded research to develop new tools to enhance surveillance and improve our preparedness for future pandemics. In this webinar, we'll hear from experts in human, wildlife, domestic animal, and environmental health. And we'll start the webinar with a presentation from Professor Gemma Geoghegan and Dr. David Winter, who are the lead investigators on the Taniwha funded project on developing new tools for wildlife surveillance. I'll introduce them first and then introduce the rest of the panel for our panel discussion at the end of these presentations. So Gemma is Webster Family Chair of Viral Pathogenesis at the University of Otago, Whakakaihu Waka. And David Winter is the science leader for genomics and bioinformatics at the Institute for Environmental Science and Research, ESR. So I'll hand over to Lucy if you'd like to share to Gemma, sorry, if you'd like to share your screen first of all, and then we'll move on to David's talk.
Kia ora koutou. Thanks, Nigel, for the introduction. So I'm just going to introduce to you a little bit about our project, and then David will take over from there. So highly pathogenic avian influenza virus, we've known about this virus for over 30 years. So why is it causing such a fuss now? Well, over the last few years, the virus has evolved and acquired genetic mutations that allow this virus to expand both its host range and therefore its geographical range as well. In fact, that we're the last remaining continent that this virus has not yet been detected on. And what's more, this virus has now spilled over to mammalian hosts, as Nigel said as well. In fact, over 50 species of mammalian hosts have now been detected with the virus. Often it is killing thousands of individual mammals at the same time. But primarily, this has been due to spillover events from direct contact with wild birds. But over the past few months, we've seen even dairy cattle across the United States being infected with this virus as well. And this has been primarily due to contact with infected poultry on farms. And now, as this virus has spread to over 50 farms across the US, there has been genetic evidence to suggest that this virus has acquired a mutation which allows it to spread between cows. So it's the first genetic evidence of mammalian transmission of this virus between cows. I must stress, though, that this virus does continue to look very much like an avian virus as of yet, though. So I'm co-leading this TNIFA-funded project with the aim of improving our preparedness in New Zealand. And there are two major questions that we think that we should answer. First of all, how might this virus get to New Zealand? And how can we improve our surveillance, given its changes in its host range, to better detect the virus when it does arrive? So, as I said, we are the last remaining continent that the virus has yet been detected on, and that means that it's even been detected in birds in Antarctica as well. So, every other place the virus has reached, it has done so via wildlife, mainly wild birds. And so, but we don't know very much about how wild birds in particular transmit viruses and how they interact between the migratory birds and the ones that don't migrate. So we basically don't know anything about how this virus might arrive and the transmission patterns that it might follow if it does arrive. So what we've been doing is working with the Department of Conservation and community groups to sample as many different wild bird species as possible, particularly across the subantarctic islands, across the flyway sites, the migratory flyway sites in New Zealand and other regions, to sample wild birds and their viruses, and then using the virus genomics to try and understand how viruses are transmitted more generally and make inferences and model how avian influenza virus might then spread across the country. Over to you, David.
Awesome, thank you very much, Gemma. So yeah, following on from that, I guess there are kind of like two arcs or two aims of our work in terms of how we can prepare, how we can use research to strengthen our surveillance toolkit, if you want. Gemma's talked about how we can use it to understand how the virus would get here and get around. And another part of it is how can we accurately detect and routinely detect the virus if we do have an incursion into New Zealand. So I think avian flus are kind of like a real prime example of a common pattern of the work that we want to do in surveillance, is that fundamentally viruses are not aware that we would like to be able to compartmentalize our lives such a way that there was a thing called environmental health and a thing called animal health and a thing called human health, and they were three separate domains. The virus is unaware of that, so it's a bird virus, but it can be acquired through the environment, and there is a potential risk for it to end up being causing human cases as well. So we want a surveillance toolkit, a set of tools basically for detection, which are fit for purpose for being able to take those different approaches to being able to detect this virus in the community, in wildlife, and in human cases eventually. So typically avian influenza surveillance is all around ducks. So ducks are a really great sentinel for avian influenza, because if there are birds in the community with avian influenza, ducks are likely to end up getting it. So they soak it up from the environment. They're just very good at getting flu. So if you go and swab ducks, that's a really useful sentinel surveillance method typically for understanding avian influenza. But it's not something that can really scale particularly well because it requires all the work of going out and finding the ducks and swabbing the ducks and sending samples to diagnostic labs and performing all the tests. So we're interested in a set of kind of emerging technologies which will allow you to take directly from environmental samples, for instance, because environmental samples are kind of like passively having trace amounts of all of the organisms which are around. So if you had like an estuary or a duck pond, from a sample of water from those places, you will have DNA or RNA information about what has visited that estuary recently. So being able to go from these sorts of environmental samples and being able to do detection either at a level of, say, quantifying all of the microbes, all of the pathogens that might be in, potential pathogens that might be in here with approaches that we call eDNA or metagenomics, which are more, again, something that we would do for surveillance as a tool for us in the labs to be performing sequencing, or for a test that can actually be kind of distributed out into the community where people can, using molecular biology tricks like these beautiful hand-drawn diagrams that one of my colleagues has generated, basically being able to use right down at the molecular level to be able to target a particular organism of interest or even a particular variant, or even right down to the level of an individual mutation that you're really interested in being able to surveil in the community. And there's this emerging set of tools basically for relatively low overhead, low requirement to be able to generate this sort of data in the field, so that you could get to the point where instead of sending samples away to diagnostic labs and performing these tests, you can do the test just by the color that a particular tube is changing. So this is a this is kind of a technological challenge. There are multiple tools available for this, and we're going to trial a few and then try and work out on a test which we can generate this sort of like what we sometimes call a point of need test, a test that doesn't require all this extra effort around it effectively. This is very much the start of the project, so we're not at the stage where we have a nice colored tube. We're at the stage where we have tubes full of duck poo and various pond water, but we're just on the start. We have the samples and we're ready to go. And then I would say, just to bring up earlier, I think the other point of this is once we have that test, that's a technical challenge, and we can build on the technical challenge, but there's another challenge, which is then once we have a test that can actually be being deployed out in the field, if you want, how do we then support the communities that are interested in what's happening in their forest and their estuary and their lake, to collect, hold, and interpret that data, but still have a national surveillance view of it. So that's beyond a technical challenge, and it's something that we're going to hopefully develop as part of this project. So I think that's our summary, and we'll move on from there.
Thanks very much, David. So our next presentation is from Dr. Rachel Stania about the work of the Department of Conservation. Rachel is a veterinarian with a background in wildlife health, both in the United Kingdom and in New Zealand. And Rachel works for the Department of Conservation in their incident management team on planning and preparedness for highly pathogenic avian influenza in wildlife in New Zealand. So I'll hand over to you, Rachel.
Okay, great. Thanks, Nigel. That's awesome. So hi everyone, I'm Rachel. As Nigel said, I'm a veterinarian working for DOC in the IMT on the HPAI preparedness. And I was asked to give a bit of an overview today of what we're doing in DOC in terms of preparing for HPAI. And then I'm going to specifically talk about the vaccination trial that we're doing in endangered species. So as we all know, preparedness for something of this magnitude really requires a multi-agency approach. And as HPAI is an exotic disease, MPI and Biosecurity New Zealand are the lead agency on this for us. But in DOC, we're working quite closely supporting them and working with a number of their staff on this project. And I just wanted to mention a few of us from the DOC IMT and from MPI as well attended a UNESCO webinar last week, and they shared some experiences with HPAI overseas, and they really stressed the importance of this One Health approach, which we all know is so important, but also the concept that we thought was vital, which is this statement of preparedness in peacetime. So where we should plan and coordinate across agencies during this phase where we don't have HPAI in the country. And it's a golden opportunity to do that across agencies and stakeholders. So we all thought that was a really great point. So at DOC, we've been working, like I say, we've been working closely with MPI, and basically our goals are to continue our important conservation work whilst reducing the impacts of HPAI and providing for animal health and welfare, and also human health and safety of our staff and volunteers. So it's quite a big challenge. And in the draft phase at the moment, what we're working on is some district plans to help guide our districts what to do when it arrives. We've got some captive facility guidelines that we're working on as well, conducting the vaccination trial, as I said, and then a large part of our work is also on communications and engagement with iwi stakeholders and third parties. So I'm just going to briefly, in a nutshell, go over what we're working on with the district plans. They're very much in the draft phase at the moment, but basically you can see what a challenge it is. We've got 49 regional districts, and each of those are very, very unique with their own set of risk profiles and challenges with respect to bird flu. So what we've done so far is basically we've done a large-scale survey of all of our districts to find out what their risks might be with respect to bird flu, what mitigations might be required, and what resources they have and don't have to help with preparedness. Based on this, we've used some international guidelines and the inventory data, and we're coming up with a bespoke plan for each district, depending when bird flu arrives. And these will help guide them in terms of where their sites and species of concern will be, what actions they should and shouldn't continue, and how they can continue with their essential conservation work safely. Also things like mitigation options that they might have in terms of site closures and carcass collections. And then obviously the next stage of this, which is really important, is to engage with our iwi council and stakeholders, and that's really part of this preparedness in peacetime that we're hoping for and to achieve. The next thing that we're working on was the captive facility document. So again, this is very much in the draft phase, but we're trying to help prepare our DOC run or DOC involved captive facilities, and we've got 4 of these sites where it's either DOC run or we're very, very involved with what they do, and they're all really vital for endangered species survival. So they're all really key in helping the recovery of some critically endangered species in New Zealand. So what we've done, again, using international guidelines, especially the zoo biosecurity tools that they have overseas, we're creating some plans so that they can develop biosecurity techniques to keep bird flu out of their facilities, and basically working out how they can continue these really important conservation activities whilst keeping the birds and keeping themselves safe, and working on preventing spillover events, so things like spillover into domestic animals. And then the final thing I was going to talk about is our vaccination trial. So this is something that we're working on in endangered species, and why we might consider vaccination of endangered species as a tool in our toolbox for preparedness. So vaccination, it reduces the clinical signs of disease, mortality rates, and environmental shedding of the virus. The purpose of our trial basically is to see if we can check the safety and the efficacy of the vaccine that we're using on a select group of species. We're using a commercial poultry vaccine, which is Poulvac Flufend, against H5N3, but is thought to have that it should have enough cross protection against H5N1, and it is a registered vaccine in New Zealand. It's not used commercially, but MPI have made it available to us, and obviously we've had to go through animal ethics approval to be able to do this. We're not the first ones to be doing it, so many studies in zoo birds overseas, and there's 2 or 3 other vaccines that have been used overseas quite a lot in zoo populations, so about 36,000 birds in total, approximately, and they seem to be very safe. There's not many adverse reactions noted overseas, But the consistent finding overseas has definitely been that different species seem to produce different levels of antibody. It seems quite variable depending on the species of birds. So we wanted to see how our birds respond. And probably the best case study for this is the Californian condor, which are very interesting. It's a very interesting situation because they have conducted a vaccination program already. And this bird went through a very severe genetic bottleneck where it was extinct in the wild, but now there's 561 birds. So back in 2023, they lost 21 birds due to avian influenza. Initially, they thought it was lead toxicity because they have a lot of problems with that, but post-mortem exam discovered that it was actually highly pathogenic avian influenza. And they have a mix of intensively monitored wild birds for the Californian condor, but also a captive population. So it was decided that they were going to start a vaccination trial, and they used black vultures as a surrogate for this initially, and then rolled it out onto using on the condors themselves, on the captive population. So they've vaccinated a fair number now, about 94 in total, 21 of which have been released back into the wild. And they've seen good responses in terms of antibody levels to the vaccination. The levels in the black vultures were higher than the condors, but they did get good responses in the condors. But the duration of antibody was quite short. So I started dropping off after about 3 months and lasted about 6 months. And there's what they look like if you didn't know. So in New Zealand, we came up with a set of criteria for vaccination. Basically, this is something that can only be done if you've got a captive or intensively managed population. So endangered species in captivity, or in the case of the kakapo, they're not in captivity, but they're flightless and on an offshore island. So we can be pretty sure they're not going anywhere. They need to be accessible and identifiable because we have to do 2 doses of the vaccine. So we've got to be able to recapture these birds, know who they are, see if they've had an adverse reaction, and take bloods for antibody levels. And we've got 3 vets working on this. The lead is Kate McInnes, and then myself and a vet called Lydia Udstrom are helping with the species vaccinations. And obviously, we definitely got local iwi engagement and agreement on this before we went ahead. So we've got 5 species that we're working with. Kakapo in the top left, we've got shore plover below it, and then takahe top right, and then the khaki or black stilt is bottom right. And in the middle, we've got 2 subspecies of kakariki. The orange fronted at the top is the most critically endangered one. They're very, very small birds, and we don't have very many of them left. I think it's about 200 to 300 on offshore islands or fence sanctuaries. So for this species, we actually decided we were going to use a surrogate, a bit like they did with the condors, and we're using the red-crowned kakariki. They're more numerous. There's quite a few in captivity, and they're larger and less prone to stress reactions. So yeah, so those are the ones that we're conducting the trial on. And the protocol, so we've got about 10 birds for each species, split into 2 cohorts. So about 50 birds in total. We decided to do a staged approach, 1 cohort at a time, just to check for adverse reactions and to check that our capture and techniques were going to be effective. We health check them on day 0 and swab for HPAI PCR, just to check they haven't got it already, and take bloods at that point as well to check and they don't have antibodies to HPAI. And then what we're doing is 2 doses of the vaccine a month apart. So, and that's given subcutaneously, and serial blood tests after that to monitor antibodies. And like I say, some of these birds are about 60 grams. So you can imagine the challenges of blood testing such small birds. Can only take about 1% of body weight in terms of blood. So there's been some logistical challenges, but so far we have all the birds have had both their vaccinations. We've had no reports of adverse reaction, and we are getting positive signs that we have antibody response, especially in takahe and shore plover are the main ones that have been tested so far. We're just waiting for results on some of the others. And I guess it's just worth mentioning that the vaccination trial And if we were to vaccinate endangered species, there really does have to be quite a specific set of circumstances with birds in captivity. It's fairly impractical to be thinking of vaccinating wild bird populations in general. So it is something that will probably be quite reserved for specific circumstances. Okay, thanks.
Thanks so much, Rachel. So thanks to Gemma, David and Rachel for setting the scene for the next part of the webinar, which is our panel discussion. The three of them will be joined by Dr. Emma Sherwood, who's a public health medicine specialist and highly pathogenic avian influenza lead for the National Clinical Protection Team in the National Public Health Service in Health New Zealand, and Dr. Nick Bonrando, who is the Chief Clinical Advisor for the Public Health Agency, Ministry of Health. And finally, Dr. Mary Van Andel, who is our Chief Veterinary Officer in the Ministry for Primary Industries. So everyone is available for answering questions. And I'll start off with a general question aimed initially at Emma and Nick, and then Mary hopefully will be able to follow up as well. And this follows on from some of the comments made by Rachel too. So responding to avian influenza bird flu requires coordination of all government agencies responsible for human and animal, including domestic and wild animal health. What are the roles played by the different agencies in Aotearoa New Zealand in terms of preparedness and response? And how do the agencies work together? So I'll start off with Emma and Nick, if you'd like to respond to that.
Sure thing. Kia ora koutou. So first off, the Ministry and Health New Zealand, we're totally on board with the One Health idea and plan. It certainly shapes a lot of the ways that we think about what our role to play is in the health sector at this particular point of the Panzootic, and how we engage with other agencies. A lot of people will probably be aware there's been quite a bit of change in the health sector recently with reforms and different structures. So this question about how our agency is responsible, if we talk about the health sector at least, the Ministry of Health now takes a very sort of strategic response, oversight, and monitoring kind of role, and we have the role of sort of convening across the health sector. Particular to HPAI, we are responsible for the New Zealand Pandemic Plan, which I think there's a question about later. We also oversee the National Reserve Supply, so these are things that would sort of come into play in the potential scenario of any human-to-human transmission. I'll pass to Emma if you want to talk about the Health New Zealand side of things.
Kia ora koutou. Yes, so Health New Zealand and the National Public Health Service in particular would be responsible for the operational response to any human or animal case of HPAI, so managing any human contacts of any animal cases. And we're also the lead for developing health and safety and IPC guidance for public and occupational groups. But as Nick mentions, we've been working really closely together alongside health, and then also with other agencies, particularly MPI and DOC, and working to kind of formalise those working arrangements at the moment. MPI are leading on the overall preparedness, so I'll maybe ask Mary to comment on that.
Thank you. Mary.
Kia ora, everyone. Thank you for that, and thanks to the speakers for their presentations. Yes, so if HPAI is detected in New Zealand or its territories, Biosecurity New Zealand is the lead agency and will coordinate any response in partnership with the Department of Conservation, Ministry of Health and Te Whatuara. So that's important there. You know, international experience has taught us, and as we've already heard today, that a One Health approach to this current circulating strain is absolutely essential. And so the surveillance, preparedness and response planning that we've touched on here are all part of our joint preparation across the various agencies. I think aside from that, you know, a range of agencies would be involved in a response to HPAI, and that would include industry groups, veterinary professionals, regional councils, organizations that support birds and wildlife, like wildlife hospitals and rehabilitators, fish and game, forest and bird, and healthcare providers. So we really are talking about a wide range of people who will need to be involved. And so at MPI, we are working with many of these groups to make sure they're informed about the possibility that HPAI could arrive in New Zealand, and that we start to take steps to prepare to keep ourselves safe across this wide remit. And then, of course, our focus across all of these groups needs to be on response options that represent the best approach for New Zealand to this particular strain of HPAI. And what we've talked about across the three agencies is that we need to focus on protecting unique native species, mitigating the impact on primary sectors, especially our poultry sector, maintain vigilance, and be there to investigate possible spillover events, and of course, taking measures to protect human health.
Thanks very much, Mary. So David, do you comments from an ESR perspective?
Yeah, I guess I would just add that, you know, ESR would be contributing to any of those sorts of situations. So we've had discussions already with the MPI Animal Health Lab from the sequencing side of things, my sides of things, from how we could support sequencing if that became part of the response from MPI. And then obviously we have you know, great depth in terms of our reference laboratories, our epidemiologists and public health physicians, and us in the sequencing team. We've been contributing to the MPHS sort of planning around this, and we'd just stand up what was needed as it came, basically.
Thanks very much. So thanks to all of you. I think we'll move on to the next question now, which is what current surveillance activities are underway in Aotearoa, New Zealand, and thinking about what new or additional scientific developments might help to enhance surveillance. So it's been touched on a bit earlier, but I think a good overview. Mary, would you mind taking this question?
No problem. So great to hear about the lab testing that's being developed, and that's really exciting to hear about. You know, in terms of surveillance, we've got some information gathering in terms of what's happening overseas, but we've also got, you know, testing and gathering of data in New Zealand. So just cover up those. So, you know, we're part of networks that are actively monitoring the spread of disease globally, and we're able to speak regularly with our colleagues in other countries to learn as much as we can about how the disease is behaving as it spreads and what effective responses look like in different contexts. You know, New Zealand is a partner in the Antarctic Treaty Area, and that's a really important place where we're exchanging information with other treaty signatories. So that's, you know, as we've talked about, that southern spread of the disease is somewhere where all eyes are at the moment. Following the, you know, we're also following with our colleagues any spillover events and in close contact with colleagues at USDA and the Chief Veterinary Officer there about their experiences with the spillover into the dairy herd there. So as we've talked about, early detection is really going to be the key to responding effectively and managing any impacts. And so, you know, our surveillance needs to be tailored towards that. One of the important things that is part of community surveillance is everybody keeping an eye out for sick or dead birds and reporting those to the Exotic Pest and Disease Hotline at 0800 80 99 66. They can be investigated and samples will go to be tested for exotic animal diseases, but obviously including highly pathogenic avian influenza. So to enhance that surveillance, we've been reaching out to veterinarians and poultry industry organizations to make sure that people are aware of the clinical signs in poultry and wild birds. And we've seen a really encouraging uptick in calls to those numbers, not, we think, because there are more dead birds, but because there's greater awareness. And that's exactly what we want to be seeing in there. Other than that, we do have surveillance of migratory wild birds and shorebirds in partnership with the Dunedin Wildlife Hospital, and they're doing routine sampling of birds under their care for avian influenza. Of course, any of these wildlife hospitals and rehabbers are also actually able to ring the exotic pest and disease hotline if there are clinical animals, and they are doing that. We have the testing kits that have gone with our DOC colleagues to the sub-Antarctic islands and to Scott Base with our Antarctica New Zealand colleagues. And so those are things where we're keeping an active eye out. There is also some export testing of domestic poultry, and those are to do with our commercial poultry arrangements. So a wide array of activities there. There's the ongoing duck sampling that I think David and Gemma talked about, and that happens in conjunction with colleagues I think it's Fish and Game help us with that one, collaboration there. Yeah, so I think that covers our animal disease surveillance that's ongoing at the moment.
Great, thanks very much, Mary. Is that either Gemma or Rachel, anything you'd like to add to that, or we can move on? Oh, okay, great. So the next question really, I guess has been touched on a little bit, but what are the most likely routes of an incursion of HPAI, in particular the new circulating strain of H5N1 into New Zealand? And I guess the other big question is there any way we can stop it arriving here? Maybe Gemma and possibly Rachel could address that question.
Yeah, as I said earlier, when we look across the world and how this virus has spread into different regions, it has always come with wildlife and primarily wild birds. And so, of course, that's the route that we're sort of, I guess, keeping an eye on the most. And the most likely sign of the virus arriving here it would be lots of dead birds, probably, and it's very hard to stop it arriving because we can't shut our borders like we did for COVID-19, for example, to migratory birds. But what we can do is adapt our surveillance systems, as Mary touched on, to better be able to detect the virus when it does arrive. So early detection, as Mary mentioned, is the key to stopping the virus's more wider transmission and potentially, stopping the destructive disease that the virus will have.
Thank you. Rachel, anything to add to that?
Not too much, just to say that there's been some work done at DOC and MPI about looking at what birds we can expect to arrive at what time of year, what migration routes that they use as well, so we know if there's outbreaks in wild birds along route that they're more likely to be at risk. And then as part of our district plans, it's going to be part of it, just helping rangers prepare for what the signs are and increasing their passive surveillance in a safe way. So, yeah.
Right. And Mary, do you like to comment on other potential pathways other than wild bird routes into New Zealand?
Sure. And as people have touched on, unlike most biosecurity threats that we do talk about here in New Zealand, human introduction of avian influenza is not the major risk activity here. But, in terms of the managed pathways that come into the country via airports and ports, these goods and items are covered under import health standards. And those standards have, been in place and kept New Zealand free of avian influenza via those pathways. So the ones that are being managed remain well managed with very strict measures. But, as we've talked about, the wild bird pathway has become the more likely route of exposure, given the way the disease has moved into Antarctica.
Great, thank you. So maybe staying with you, Mary, if there was an incursion, a wildlife incursion into New Zealand or other animal involvement, what would be the response of Biosecurity New Zealand MPI to that?
Sure. And so as you've already picked up on in your question, Nigel, any response to HPAI and the actions will really need to be guided by what the infected species is and the location. So we'll need to react differently to an incursion detected first in wild birds than one detected in commercial poultry. Our focus is on response options that will have the best outcome and best approach for New Zealand to protect the native species, mitigate impacts on primary sectors, and carry on with investigating spillovers and protecting human health. So as with any infectious disease control program, we'd be looking to find infection early, contain that, stop it from spreading, and then implement the relevant and useful control actions. And those will depend on what the species is and the location. I know the poultry industry are learning from their counterparts abroad about operations on farm, and we can continue to learn and evolve our planning in alignment with those countries that have been dealing with the strain of the disease. Certainly had some interesting information and fruitful conversations with our industry partners in the cattle industry in relation to how cattle spillover events might be handled in New Zealand if that situation ever did arise here, what legislative frameworks we have across the different sectors, and how we would use our laboratory arrangements. I think one of the most important things that we are going to have to get to grips with here is making sure that good biosecurity practices are in place between farms and on farms, because limiting the spread through every pathway that we have in our control is really what we have to do. And that will mean, those fomite spreads, so equipment, transmission with clothing or boots, those types of things, those need to be mitigated strictly, and then also decreasing contact between wild birds and domestic stock.
Right. Okay, thanks very much. The next question really for Emma and Nick is, how would the health system respond to the first human case of highly pathogenic avian influenza in Aotearoa, New Zealand?
Kira, so obviously planning is well underway for this across all agencies, but specifically for health, we're developing a health sector interception response plan and operational guidance for public health services. And we've got expert working groups established who are feeding into all of those with a One Health approach. But ultimately, a human or indeed animal case would be recognised as being of the highest priority for all agencies. So we would be standing up a national incident management team and a clinical threats advisory group to provide expert guidance. As I mentioned, National Public Health Service would be leading the operational response, and there we're focusing on rapid detection and containment, and we're working very closely with our colleagues in Ministry of Health, MPI and DOC. In terms of kind of the detail of what we'd be doing, it would be managing the human case, so treatment with antivirals, isolation, and then public health services would be undertaking contact tracing, risk assessing, and management of contacts. So looking at things like active follow-up, checking in with contacts daily, providing information, considering giving antivirals to close contacts of a human or animal case, and testing them. So what would be doing would be standing up enhanced surveillance, looking at people who are exposed to the environment or exposed to the cases, testing them to see if they are testing positive for H5N1. And trying to gather as much information as we can in that way. And then we'll also be using some other kind of routine measures like influenza surveillance, which we do routinely to be looking more widely. And finally, of course, IPC, so PPE and mask wearing and basic kind of hygiene and sanitation measures for anyone managing the outbreak would be really key, alongside reassuring the public that the risk currently would be negligible if they're not exposed to infected animals or animal material.
Thanks very much.
Yeah, I just add that yeah, in that situation of a sort of sporadic human case, I mean, a lot of these measures and a lot of the response are essentially there's a bit of precautionary principle to them, because we've never seen, particularly of this clade, any human-to-human transmission, certainly not sustained. But we absolutely recognise that there are concerns about high case fatality rates, and that there's a great degree of risk perception. There is also that potential scenario in which a human case arises without knowing about any wildlife or animal cases in New Zealand, and we've got a particular responsibility to quickly find out whether that's likely to have been acquired in New Zealand, essentially, or whether that's a sort of imported case that may have come from overseas, because we recognise that there's very probably pressing and major implications for finding it, finding out that information and being able to communicate that with our One Health partners.
Thank you. I do want to get on to a couple of the audience questions, but just before we do that, a couple of, we could get some quick responses. Mary, you've already touched on what the public should do. Maybe Rachel, do you want to expand on what the public should do to help surveillance activities and protect themselves in terms of identifying any outbreaks, say, in wildlife, for example?
Yeah, thanks, Nigel. So our advice generally would be to look out for any unusual signs in wildlife, mostly respiratory, neurological, if they find more than 3 dead wild birds, and also not forgetting about marine mammals too. They've been quite heavily affected in other countries, especially South America, so not just wild birds, but marine mammals too. Things like keeping a distance, not touching the birds, ringing the Biosecurity New Zealand hotline straight away to report it, and also keeping their dogs away if it's on a dog walking beach, things like that, making sure scavenging can't occur. So those are sort of key messages we've been working on.
Thank you. Mary, anything to add to that?
No, thank you.
Great. Another question that's often asked, again, if we could just get a brief response to this, is how does avian influenza or bird flu differ from COVID-19? Given our recent experiences, I think this is an important thing to address. Maybe Emma, Nick, would you like to start with that one?
Yeah, sure thing. I think one of the major differences is that most of the world has been sort of living with this virus for this particular strain of the virus for a couple of years, but this slightly broader whānau of viruses for decades. So we tend to know a lot more about it, which can be quite helpful. There's certainly reports of a higher case fatality rate than COVID, although any of those reports do need to be interpreted with the knowledge that we probably don't know everyone who's been infected, so it's probably a bit inflated. And a lot of the mortality that's happened has been in countries with health systems that aren't necessarily comparable to ours. We have the National Reserve Supply, and we have candidate pre-pandemic vaccines that are there, that obviously they haven't been through rigorous human trials because there haven't been enough people infected, but there's laboratory evidence that they're likely to have some effect. And also the world knows how to make influenza vaccines fairly well. So it's a different situation from COVID, where we started out really not knowing where the future in vaccines and treatments was going to be. We also have effective antivirals as well. So there are a lot of differences that mean that it's a fairly different situation. But obviously we're not completely relaxed about it. We're vigilant. We're monitoring the situation, and we're doing all that we can to prepare for some more serious scenarios.
Thank you. Thanks. I think we'll move on to questions that we had sent in from the listeners. So the first one from Michael Baker is, what is New Zealand doing to update its current influenza pandemic plan to incorporate lessons from the COVID-19 response? Again, I might throw that one back to Emma and Nick.
Sure thing. Kia ora, Michael. Before COVID-19, the main health planning document for a pandemic was the New Zealand Influenza Pandemic Plan. That was last updated in 2017. It was a valuable resource during the COVID-19 pandemic, although there were certain things that it didn't necessarily envisage, like the degrees of lockdowns or managed isolation and quarantine. Our understanding of pandemics has obviously evolved significantly since 2017, because we've had a lot of real world experience with that. The other thing is that we've had the health system reforms, so roles, responsibilities, structures, and legislation have all changed, and it's a new operating environment. So the ministry has been undertaking an interim review of the pandemic plan to reflect those changes, and some of the early lessons of the COVID-19 response. So that's the first phase. There's going to be a fuller and more comprehensive phase that's done after that, particularly dependent on the findings of the Royal Commission of Inquiry for COVID-19. So we're really awaiting the results of that, and for that process to work itself through before we officially incorporate all of those things into New Zealand pandemic planning. We're also going to make it somewhat more disease agnostic, so that it's not just an influenza pandemic plan, because obviously a lot of the lessons from influenza were applicable to COVID-19, we do need to think about some other types of particularly respiratory pathogens.
Okay. Thank you. Thanks very much. The next question is really for Mary, I think, from former colleague Matt Stone. So how are pigs likely to be involved in future avian influenza scenarios? Do outdoor pigs with potentially low natural immunity to animal flu viruses, but some evidence of exposure to human viruses, increase the public health risk profile? Is this exposure risk being considered, and could industry comply? So quite a long question, but
You're a Matt. I expect nothing less. Yes, so I mean, I think like many people, we've been watching the various spillover hosts that have been identified in other countries with interest, and I think it's fair to say that they've in some cases been unpredictable. So regardless of the fact that up till now, spillovers into domestic pigs haven't really been a feature of international reporting, we're still, being conservative, working with our colleagues in the pork industry, to talk about these biosecurity measures, talk about potential spillover events, talk about what the process for sampling and testing might look like. Certainly housing orders for poultry have been a successful tool in the toolbox, and they would need to be considered as biosecurity measures in the New Zealand context. So certainly on our radar, and working through some of those details. I don't have the answer about the increased public health risk profile. I think I'll refrain from that part of the question, seeing as I'm not the relevant expert for that one. Thank you.
Well, I think that question might I don't know, Gemma, do you have any comments on that? Pigs as mixing vessels with combination of human and other viruses?
Yeah, pigs have been really important in the emergence of other flu strains in the past. We've seen, for example, in 2009, the swine flu pandemic emerged from pigs, and pigs with their ample ecological, contact with poultry as well as humans seem to be a really good mixing vessel. Their upper respiratory system seems to allow the virus to replicate in a way that it would provide it a really good source of genetic mutations that would allow it to also replicate in the upper respiratory tracts of humans as well. So they seem to be very good in terms of allowing the virus to adapt for mammalian transmission. So I think a lot of the world is very worried about that scenario unfolding, and if pigs become a reservoir host, and the potential of the virus more likely to becoming human adapted.
Thank you. We have a question from Catherine Wolfe from New Zealand Geographic. Given the understandable limitations to vaccination, I'm interested to hear which wild native species populations among those that cannot be vaccinated are considered most vulnerable, and are there types of work underway to help protect some of these birds? And there's also a sub-question around vaccinations available for endangered mammals, e.g. sea lions. So Rachel, are you able to respond to that one?
Yeah, sure. So we are currently reviewing what species might be reconsidered for vaccination. Should the trial be successful and prove to be safe and work. So that's sort of next steps. And we're also working on some species-specific mitigation plans, so to work out what other steps can be taken. I think one of the big things that we've sort of learned from overseas where populations are recovering is one of the main things is trying to reduce other pressures on these species. So, you know, things like if we've got predation from invasive pests, then we need to try and make sure that we're continuing to reduce these other pressures on these species to help them with their immune system, to help them cope with surviving HPAI. So a lot of the mitigations are going to be around those other things we can do, not necessarily vaccination. Vaccination might be something that we employ if we're having to take the remaining number of a certain species into captivity and manage them in that way. I'm also not aware of anywhere where they've used the vaccine in sea lions at this stage, but something to look into.
Right, thanks, Rachel. And the final question from the audience from Professor Graham LeGros from the Malagan Institute around rodents. So rodents, do they pick up bird flu? Is a specific question around hamsters as well. I don't know who's the expert on rodents and bird flu.
I think there's no reason why not. There's been, the updated is the 60 mammalian species that have now been infected with it. Definitely mustelids seem to be a big one. And I think one of the key things is trying to work out what the pathways are for the mammals to be infected. So, you know, things like these pest species getting into seabird nests, etc. I can't see a reason why not.
Gemma, did you want to add to that as well?
Oh, I mean, what Rachel said is correct. And rats have also been one of those mammalian species on that list that have been infected before. And I'm sure other rodents would be as well.
Okay, great. Well, thanks very much. So we're just 2 minutes from the hour up now. So I think we've managed to answer most of the questions that we intended to ask during the webinar. So it just remains for me to say a huge thank you to the presenters and the panel members for participating in the webinar and providing such insightful commentaries on what is an emerging and very rapidly evolving global situation at the moment. So Ngā mihi nui to all of you, and thank you very much to our audience of listeners as well. And we'll sign off now. So ka kite.